Assalamualaikum and hi everyone. This is our video presentation for the subject CSI 162. Our topic is laboratory analysis technique on hot press soap. Let me introduce our group member. First person is Aimi INANI binti Zairul Hisham. Second is RAIHAN SYAZWANI binti Maisam. Third person is AINUL KAMILAH binti Abdul Wahab. The fourth person is Nur Dalili Batrisyiah binti Zainal Fitri, and the fifth person is Nur Najwa Najihah binti Muhammad Aizal, and the last person is Nur Zalika Zahra binti Zainal Abidin.
So, the laboratory analysis of hot press soap is really helpful to check its quality and make sure it's safe to use. We do tests like measuring the pH, moisture content, and checking the ingredients. This helps us confirm that the soap meets the right standards. These tests are also important to improve the product so it works well for users.
Problem statement. First is daily exposure to harmful statement. Various exposure to microorganism on daily basis that can transmit disease. Example e.g. Aeruginosa, E. coli, and S. aureus. Second is limited use of natural ingredients in soap product. Some hands or body soap product in the market have not been clinically tested, which can cause short or long-term effect to the user. Lastly is lack of clinical testing in some commercial soap. Less body soap product has been introduced and marketed using this natural product.
Methods of analysis. Method of analysis used for this experiment is soap analysis of pH using pH meter, weighing methods using an analytical balance, and microbiological analysis using nutrient agar plates. Soap analysis of pH using a pH meter. pH meter is used to measure the alkalinity of hot press soap, ensuring the pH of soap falls within an acceptable range, which is crucial for ensuring the soap is safe and gentle for skin use. Firstly, the tube containing hot press soap was placed in the beaker. Second, the pH probe was carefully inserted into the tube without touching the sides. This is to prevent contamination and interference with the measurement. Third, the reading was allowed to stabilize for several seconds before being recorded. This is to ensure result accuracy because the sensor of the probes takes a few seconds to adjust to the sample's condition. Lastly, once the reading stabilized, the results were recorded. Hence, the reading of the result is 10.91.
Now, let's move on to number 2, weighing method using an analytical balance. In this method, an analytical balance was used to determine the mass of small soap sample with a high degree of accuracy and precision. To begin with, the analytical balance was placed on a stable surface and turned on. Next, a weighing container was placed, and the tab button was used to zero the reading. Then, the soap was added carefully using a spatula. The final value was recorded once the display becomes stabilized. And finally, the balance was cleaned properly after use. This process ensured that our measurements were both reliable and accurate.
The third method is microbiological analysis using nutrient agar plates. To evaluate the antibacterial effectiveness of the hot press soap, we conducted a simple hand swab test using nutrient agar plates. First, a sterile cotton swab was used to collect bacteria from the surface of an unwashed hand. The swab was then streaked across the surface of the first agar plate, labeled plate 1. Next, the same hand was washed thoroughly with the hot press soap and dried using a tissue paper. After that, a new sterile swab was used to collect bacteria from the washed hand, and the sample was streaked onto the second agar plate using the same technique. Both plates were incubated at 37 degrees Celsius overnight to allow bacterial growth. After incubation, the results of both plate 1 and plate 2 were observed and compared. The third method is microbiological analysis using
This experiment was conducted to test the effectiveness of soap on bacteria using two methods: hand wash without soap and hand wash with hot press soap. The stage was observed before incubation and after incubation. Without soap, before incubation, the plate looked clear, and after incubation, the plate stayed clear and no bacteria grew. With hot press soap, before incubation, the plate looked clear, and after incubation, the plate stayed clear and no bacteria grew. This experiment was conducted to test how effective hand washing is without soap and with hot press soap. Both methods show no microbial growth on the agar plate, which means the good accepted technique were applied and low environmental contamination were present during the experiment. For hand wash with hot press soap, the soap is helped in remove or reduce the number of microbes. This shows that using soap is important for better hand hygiene and from prevent the bacteria growth. The good hand washing technique were applied for both methods. The proper hand hygiene using hot press soap is effective in removing microorganisms and from preventing the contamination.
In conclusion, lab analysis on hot press soap help us check its quality, safety, and effectiveness. It ensures the soap meets the right standards and is safe to use. These tests also help improve the product for future use.